just introduce myself. I'm Will Gibson. I'm a medical oncologist at Dana-Farber Cancer Institute, and I also run a chemical biology lab there. Today I'll be talking about some induced proximity approaches to cancer therapy. I want to thank an amazing undergrad I worked with and my two former mentors, Matthew Meyerson and Stuart Schreiber at Dana-Farber and the Broad Institute, and my clinical mentor, Steve Hody. The goals in my laboratory are to develop new chemical platforms to rewire cellular circuitry such that we can create new cancer therapeutics. And these include bifunctional molecules that can enable targeted protein relocalization, technologies for targeted transcriptional activation as well as repression, and to use these platforms to address some of cancer's most intractable targets, such as drugging p53. So one way to think about how this approach works, or how kind of everything in biology works, is that proximity underlies everything. So imagine you have this simple chemical reaction here between this acid and this acid chloride. It goes very slowly, actually, in solution. But say that you link the two components together covalently, then this reaction goes fairly quickly. Now, keep in mind that this here is a rotatable bond. If you then constrain the elements such that they are oriented favorably, the reaction goes very, very quickly. So in chemistry, lining things, there's no magic. It's just lining things up very closely allows reactions to go quickly. The same thing happens with biology in nature, which is that if you take these three proteins, they really don't do anything together if they're not oriented correctly. But in yeast, there's this scaffold protein called STE5 that orients the three proteins together such that the chemical reactions actually occur. So proximity underlies every process in nature, but maybe we can use those things in our favor if we orient things in the ways that we'd like them to go. And what's happened over the past decade or so is that there's been a paradigm that has emerged, which is if you take a protein of interest and an effector enzyme, and you design dumbbell-like molecules that bind to the two of them, you can enforce their co-proximity such that post-translational modifications occur at a rate where they normally wouldn't occur in nature. And you can do the same thing with removal of post-translational modifications. So if you look towards nature, we find that nature employs molecular glues, so small molecules that induce neo-protein-protein interactions that don't typically occur. So one of the first ones that was described was FK506. It's a natural product that binds to FKBP12, and in so doing, the ternary complex can then bind to and inhibit the key T-cell protein calcineurin. Cyclosporin is an analogous molecule produced by bacteria that binds to cyclophilin as a presenter protein, and then that complex of cyclophilin and cyclosporin can then bind to and inhibit calcineurin. And perhaps the best known of these small molecules is rapamycin, known for its effects on T-cell inhibition and longevity, which binds to the presenter protein FKBP12, and the ternary complex can then bind to and inhibit the kinase mTOR. It used to be thought that these molecules were evolved by billions of years of evolution and couldn't be recapitulated synthetically, but over the past decade we found that we can do that in a rather facile way and prospectively too. But the point is, nature uses molecular glues to create new protein-protein interactions. So many of us are familiar with this thing called PROTACs, which is if you design a small molecule that binds to a ubiquitin ligase and a protein of interest, you can induce the polyubiquitination of the protein of interest. And this is Craig Crews and Ray Deshay, who discovered this at Yale in 2001. It became known more recently by Ben Ebert at the Farber and Bill Kalin, who won the Nobel Prize, that one of the best-selling drugs in the world called lenalidomide actually worked in this way. Lenalidomide binds to the cereblon ubiquitin ligase, and then that complex can then bind to and inhibit and degrade, actually, the Akeros transcription factors, which are essential for myeloma cells. So at the time, lenalidomide was the #2 best-selling drug in the world. It sold $8 billion a year. No one knew how it worked. But these experiments showed that these molecular glues that induce these new protein-protein interactions actually underlie some of the most successful medicines we have in medicine today. So, so there's this observation that you can degrade proteins of interest if you induce proximity with ubiquitin ligase. And well, again, the idea came up with, is this just like a trivia fact? Is this a curiosity of nature? But then it turned out that Jay Bradner, who then was the president of Novartis and now is the CSO of Amgen, showed that you can copy and paste this lenalidomide, the cereblon binding small molecule, to a binder of a protein of interest. Here, this is JQ1, which binds to the bromodomain protein BRD4. And in so doing, you can create highly effective degraders of the protein of interest. So you bring something to the trash machinery of the cell, the trash machinery ubiquitinates it and destroys it. So the old model of pharmacology was a occupancy-based model where you have an enzyme and a substrate and you design a competitive inhibitor that blocks the substrate's accessibility to the enzyme. It's how we think most drugs work, right? But the new model is a proximity-driven model of pharmacology where a small molecule induces the proximity of two proteins and then one protein performs an action onto another, in this case ubiquitinating the IKZF1 transcription factors. So that was kind of the era from 2014 to 2019, but I think the era from 2020 onwards is how can we use induced proximity beyond just degrading proteins? And today I'll show you a few vignettes about how my laboratory is working on that. So we've seen across the world, this has been used for phosphorylation of proteins, dephosphorylation at Genentech. Carolyn Bertozzi, who won the Nobel Prize, has shown this for extracellular targeted protein degradation, and Matt Disney in Florida has shown this for compounds that can even degrade RNAs. So the summary here is that bifunctional molecules can bind the unbindable and can confer neofunction, which is that they can induce degradation, phosphorylation, et cetera. And the question is, what new functions can we unlock? So I wondered this in probably around 2021 and sought to develop a project on, could we relocalize proteins using enforced proximity? So this is one protein grabbing another and pulling it through the nuclear pore. And just as an example, we designed a small molecule. Here you can see this protein starts out in the cytosol of this cell, and when we add our compound, it tracks to the nucleus within about 10 to 15 minutes. So we designed some small molecules that bind to cytosolic proteins here in green, and here's the green binder. It's a compound called AP1867. And then we appended to it, through a little linker here, a compound that binds to the highly abundant and constitutively nuclear localized proteins BRD2, 3, and 4. And we hypothesized that if we enforce the coproximity of these two molecules, either the red protein is going to move to the cytosol or the green protein is going to move to the nucleus. And what we found is that in so doing, when we added our compound, we were able to relocalize the cytosolic protein, here GFP tagged, to the nucleus within about 20 to 40 minutes, and it becomes really binary. We wanted to create a cleaner system, so we used a lentiviral infection to do it, and we were able to relocalize the compounds very nicely to the nucleus as well, using endogenous BRD4. If you cut the binder here at this little linker, then the unlinked components do not have this relocalizing activity. And here's just a quantification of that, which is that only in the intact compound works. And you can compete off this effect by adding an additional amount of either of the monovalent binders here. So we added more of the BRD4 binder with JQ1, and we're able to do the same thing with AP1867, which is you see localization at low concentrations of the monovalent binder, but when you add more monovalent binder, you ruin the effect. So a further feature of these dumbbell-like molecules is that at low concentrations, you don't induce any ternary complex formation. And at moderate concentrations, you cause the association of the two proteins. But at very high concentrations, you compete off both sides of the ternary complex formation. And this kind of dumbbell-shaped curve of activity is called the hook effect. So we show that when we add moderate doses of compound, we get nice nuclear localization, and when we add very high doses, we get the hook effect, which is what you would expect from these dumbbell-like molecules. So we did some experiments to show that we could apply this to a protein called phosphoinositide 3 kinase, which is involved in cancer, and I'm going to kind of skip through that part. But this is a version of a cancer-causing protein called NPM1C where it's constitutively nuclear localized due to mutations at the C terminus of the protein. And the idea is that if you could relocalize it to the nucleus, that could be an effective cancer therapy. So we showed that using our sort of paradigm, you can relocalize the compound to the nucleus really nicely within minutes, and that there's some single-cell heterogeneity in how this happens. I'm going to go a little bit ahead just because we're limited on time here to tell a story about how we can use some of these technologies for a gene called P53, which is the most mutated gene in cancer. So P53 is the most mutated gene in cancer, and it's not by little, it's by a lot. About 35% of patients have P53 mutations. And of the patients who develop cancer, so here's a profile of various different histologic, or I should say genomic subtypes of acute myeloid leukemia. The patients with P53 mutations here in purple are those patients who are the ones who actually die of their disease. So two forces conspire to make P53 just so important for cancer. One is that it's the most common genetic alteration, and two, it's the worst mutation to have. So adding these two things together, about 5 million patients with P53 mutant cancers die every year of their disease. So if we could move this figure by just even 1%, it would be a good way to spend our time, let's just say. So what does P53 do? It's a tetrameric transcription factor, meaning that 4 different protein copies have to come together in space. They have to bind to DNA in just the right places across the genome and then transcribe genes involved in cell death. So the way that it looks is like the tetramerization domain is here in red, and it's almost like you have a bunch of different meatballs on strings of spaghetti all tethered together. Now, the places that P53 binds in the genome are these things called half sites. So one dimer of P53 proteins has to bind to this half site, and the other dimer has to bind to this half site. And it has to recognize exactly those places in the genome, or else it's in the wrong place, and it's not going to transcribe the right things. So it's as though nature just found out how to make cells kill themselves once, and then it hooked up all the machinery to it. So if the cell were sort of the semiconductor, you'd imagine all these different little signal integrators all passing into P53. And what cancers like to do is they like to disable the P53 output. So most tumor suppressor genes like APC or ATM or BRCA1 are disabled through frameshift mutations in yellow or nonsense mutations in blue here. So you would expect the same thing of P53. But actually, most P53 mutations are these missense mutations, 75% here in green. And the reason is that these mutations poison the tetramer. So most of these mutations occur in the DNA binding domain here and at these certain hotspot sites. So here's the prevalence of the mutation, here's the gene body. And if you look at the places where the mutations occur, they occur at these positively charged arginines that interact with the negatively charged backbone of the DNA. And so if you disrupt that Coulombic interaction, then you disallow DNA binding at the correct consensus sites. Now, if you imagine assembling tetramers from a wild type genetic copy and a mutant genetic copy randomly, then only 1 out of 16 of those tetramers will actually have all of the wild type components. So a single missense mutation of P53 destroys 15/16 of P53 function. And so cancers develop these hotspot mutations rather than these disabling nonsense mutations because it's a faster evolutionary path. You just need a single event for it to happen. So one had hoped that one would be able to design a binder to, an inhibitor of, or a degrader of some other protein that existed in the cell, and that would have a specific effect on P53 mutant cells. So we went and looked for such proteins by doing genome-wide CRISPR screens across thousands of cancer cell lines over the course of the last decade. And unfortunately, nature never owed us that favor. So here's the dependency score of the wild-type cells, here's the dependency score of the mutant cells, and you can see it's kind of all on the same axis line. But the things in the P53 pathway, and if something was here a synthetic lethal, so to speak, it would be down in this corner, and it's empty, unfortunately. But we have massive statistical power. Everything that is in the P53 pathway falls off of the identity line, like its negative regulator, the ubiquitin ligase MDM2. So the conclusions are that we have massive statistical power to find synthetic lethals, but they just don't exist. So if God came down tomorrow and gave us a degrader of every protein, we'd actually be no closer to solving how to drug p53. So then you ask, what is different about p53 mutant cells versus wild-type cells? And you see that there's only one difference. This is every protein, the abundance of every protein in the proteome from global proteomics. Here's the p-value on the y-axis. Only one protein stands out, and it's p53 itself. The reason for this is that p53 protein here on the y-axis, measured by reverse phase protein array, accumulates massively in cells that have missense mutations here in yellow compared to nonsense mutations versus wild-type cells in gray. And a picture is worth 1,000 words, so here's a patient sample. This is hematoxylin and eosin staining on the left of an ovarian cancer patient with a p53 mutation. These are the cancer cells. These are the normal surrounding cells. And here's the adjacent slide with p53 staining. You can see that the ovarian cancer cells are just chock full of p53 protein, and the normal cells have essentially undetectable protein. I mean, this is so much so the case that 50% of patients with p53 missense mutations have circulating autoantibodies against p53 because these cancer cells that are so full of the protein, when they lyse and die, expose the immune system to an antigen it hasn't seen before. So the interpretation here is that we would be able to specifically drug p53 missense mutant cells if we had a drug that killed cells in proportion to p53 protein abundance. So it's almost philosophically as though one were doing some soldering to reconnect the p53 output, because everything up here is working in the signal integrator, down to the cell death machinery. So I had tried a lot of things in my postdoc to try to make a therapy that specifically took advantage of p53 protein abundance, but I saw this paper from Craig Cruz at Yale that had this really cool paradigm, which is that if you have an accumulated intracellular protein called the target protein in purple here, and you design binders to it and hang off of those binders an inhibitor of a cell essential protein, pink here, you can actually achieve cancer-selective cell death. And all that you need is an overexpressed intracellular protein, of which p53 is a great example. So I look for proteins that are highly essential to cells. The essentiality is on the y-axis by CRISPR genome-wide screens. And then the abundance is on the x-axis here. So I wanted things that are not very abundant, but highly essential, and that have high-quality small molecule inhibitors. And this is the protein that popped out to us, which is polo-like kinase 1, and Boehringer Ingelheim had generated some really high-quality chemical probes that had a functionalizable nitrogen here. So we created a toy system where we overexpressed p53 mutant in yellow, along with a fluorescent marker M-cherry, and a covalent an enzyme that forms covalent adducts with these chloroalkyls here, called the halo tag. And when we did this, we were able to show that we were able to kill the p53 mutant cells at about 100-fold lower concentrations of this compound than the parental cell line. And here's a crystal violet staining, and here's what it looks like under the microscope if you look at a plate of these cells in a petri dish. We did a competition experiment. You mix the cells together 50-50, and after a week of just control, you get 50-50 concentration out. But if you add our toy compound, then you're able to fully get rid of the p53 mutant cells. And I know we're running out of time here, so I'm going to just skip ahead. These are some other controls, but we went into the patent literature and found some real p53 binders. So this is a binder to a particular mutant of p53, this Y220C mutant. And I was able to see that this mutation creates a little ledge on p53. So the tyrosine that normally sticks out is replaced by a much smaller cysteine, and that creates a little neo-pocket to which one can develop small molecule binders. And in their patent, they showed a little bit of structure-activity relationships, and we could see that this portion of the compound kept changing. They kept playing with it. And they also showed some nice melt curves to show target engagement. But finally, it was really cinched for us when we saw crystal structure, and we saw this piperidine being exposed to the solvent, which showed us, one, what part of the compound was exposed for functionalization, and also that we could use this nitrogen here. So we created a bifunctional compound enacting this approach, and put the Boehringer Ingelheim PLK1 inhibitor on the other side. And it worked right out of the box, that we were able to induce selective p53 selective cell death at very drug-like doses, 30 nanomolar, 300 nanomolar with this compound that we called p5301. We did some other controls. Neither the parent compound PMV6 nor the PLK1 inhibitor has any effect on p53 mutant cells, and only when you link them together do you get this effect. And it has no effect on other mutations that do not bind to the p53 binder that we synthesized. We did some biophysical studies to show that you get ternary complex formation. So here's a system where you express the two components and you create a split enzyme that creates the firefly enzyme that makes them glow at night. You split it in two parts, and when those parts come together, you get photons. And so we see the emission of photons when we add our compound. And finally, I'll just kind of wrap up here. So our compound doesn't work by turning p53 on. It actually works in a totally different way. It works by inducing mitotic catastrophe. So we can see here that when we add our compound, we retain cells at the G2M transition. We did a little bit of, so this is, we do see that if you incubate cells with our compound over the course of 3 weeks, a p53 low population emergence. So we do see the emergence of resistance. And we did a little bit of linkerology using 9 different linkers, and we were able to improve the potency of our compound from the 300 nanomolar range down to the 30 nanomolar range. So here are the conclusions, which is that p53 protein is massively elevated in p53 mutant cancers, that p53 protein abundance can be leveraged as a therapeutic vulnerability, and that early data are that mutant p53 can be repurposed rather than reactivated. And in the future, my lab's working on finding more cooperative compounds that glue mutant p53 to other cell essential proteins. So I will stop there and take any questions. Thanks.
Right over here. I'm just interested in the it's not my area of expertise, but your talk has inspired me to ask about the supramolecular chemistries involved in these triads. Is it usually hydrophobic interaction, or is there pi and other? I would assume that there is everything. Or what do you know?
Yeah. So usually the way you can get these proximity-inducing compounds is you take 2 compounds that bind in hydrophobic pockets, typically, and then you just link them together. And the dumbbell strategy really works. I think where the magic is how do you create compounds that are more drug-like and not dumbbells, that are like true molecular glues, where a lot of the protein-protein contacts are giving you some of the biophysical forces for affinity. And I think companies like, what's it called, isomorphic labs, like the ones who did AlphaFold, they're really at the edge of solving this, actually.
Yes, I have a question. When you show that after, I think that's 7 days, a little bit of p53 come back.
Yeah.
Yeah, so what did you try to do then, like treat again, or?
Yeah, I think that, so my strategy relies on the cells having a high abundance of p53 protein. That's the vulnerability we're exploring. I think that what we're seeing even very quickly is that cells can dispense with p53 protein rather quickly. So I'm mostly fessing up to the concern rather than saying how we solved it.
Uh-huh.
I think it's just going to be, resistance will be an issue we'll have to watch very closely.
Yeah.
Yeah.
And did you try to move from cells to organs, culture in organs or something like this, more like a tissue culture, and see if,
Yeah, I think we'd like to move into,
organotypic? No.
to real sort of animal models and stuff. We have to do some PK/PD to make sure that the compounds last long enough in the body that they don't get just chewed up by the liver right away. So there's a lot of work left to do that.
Yeah, because it's a very interesting point, I think, that you made here. So,
Yeah.
now you see, I know because I work in the past in culturing, and then just try to move from the animal, but it's very
Yeah.
good, very good. So, yes.
All right, thanks, guys.
Thank you, William, for the talk.
Thank you so much.
Yeah.